Hello, I'm Jacquelyn Meisel, a postdoc in Mihai Pop's lab at the University of Maryland. My poster is titled Assembly Graph-Based Variant Discovery Reveals Novel Dynamics in the Human Microbiome. My research focuses on characterizing the microbiome using whole metagenomic shotgun sequencing. More specifically, I'm interested in looking at microbial strain-level variation using metagenome assembly graphs. Our lab developed a tool called MetaCarvel that uses paired end read information to orient and link contigs together into longer scaffold sequences. MetaCarvel generates graphs where each node is a contig and edges denote sufficient mate pair support between contigs. Unlike other scaffolding tools, rather than outputting a single optimized sequence for each scaffold, MetaCarvel also looks for structural patterns in these assembly graphs that may provide biologically relevant insights. For instance, MetaCarvel can detect bubble structures, which manifest when contigs diverge into one or more paths before converging again. Triangular bubbles can indicate the insertion or deletion of a sequence, while bubbles with two diverging paths represent species or strain-level variants in a sample, and bubbles with three or more multi-node paths represent more complex strain-level variation. We used MetaCarvel to scaffold the assemblies of 934 human microbiome project samples from 6 different body sites and identified over 9 million variants within the samples. We next looked to determine how many insertion-deletion events and strain variants were identified in the gut compared to the oral microbiomes. We realized that the differences in sequencing depth between different body sites would strongly bias our results, so we developed an algorithm called ANCHOR to compare bubble variants across samples. ANCHOR reduces bias by using statistical models to determine the mean number of variants that would be detected if reads from one group were downsampled to the average sequencing depth of another group. ANCHOR calculates the probability that each edge in all bubbles would survive at a lower depth of coverage and retains edges with sufficiently high probabilities. Once edges with low probabilities are removed, ANCHOR determines whether a bubble would be retained at a lower sequencing depth. Here, on the left, we enumerate all possible combinations of how edge removal could affect variant structures. ANCHOR runs multiple iterations to obtain bounds on the number of bubbles of each type you might expect to see at a lower sequencing depth. We used ANCHOR to normalize structural variance of all samples to the cheek samples and found more insertion-deletion events and strain variants in the oral cavity microbiome than in the gut. We hypothesized that this may be due to nutrient availability, because the gut, especially stool, which is a composite of digested food moving throughout the intestinal tract, contains more nutrients than the mouth. When nutrients are sparse, microbes likely need to develop different strategies to survive, leading to higher diversity even within closely related strains. Here are just 2 examples of complex bubbles found in stool samples. In the first uppermost bubble, we can see species-level variation within Bacteroides. In the lower variant, we find contigs annotated as both Roseburia and Eubacterium species. While this first may seem like an artifact, these organisms are all butyrate-producing bacteria from a phylogenetically related Clostridium cocoides cluster, highlighting MetaCarvel's ability to find structural and functional variation that may not be detected if using reference genomes. In order to compare variation across the oral cavity, we co-assembled reads from the tongue, cheek, and plaque of a single subject. We identified many structural variants, including the Streptococcus strain variant shown here. When we mapped sequencing reads back to the contigs in this variant, we found that reads from the cheek predominantly mapped to the upper path containing Streptococcus pneumoniae contigs, while reads from the tongue sample mapped mostly to the lower path with a single Streptococcus mitis contig. Furthermore, the path favored in the cheek contains a bacteriocin gene that generates an antimicrobial peptide. This peptide inhibits cell wall biosynthesis of closely related species, thus preventing colonization by potential predators. This suggests spatial differences in secondary metabolism in the oral cavity, and also suggests that despite the close proximity and shared nutrient supply of these two oral environments, bacteria may sometimes utilize different mechanisms to establish or maintain colonization at each site. In conclusion, metacarval is a method for scaffolding and variant discovery in shotgun sequencing datasets. Compared to alignment-based methods that rely on reference genomes and can only identify single nucleotide variants, graph-based variant detection allows for resolution of sophisticated functional and structural changes in mixed microbial communities. Metacarval identified more than 9 million variants in the HMP samples, including strain-level variants that suggest ways in which microbes may adapt to or colonize different niches. Metacarval is available on GitHub, so please check it out and try it on your own sample.